Hi, I'm Uli Mathesius. I'm a plant scientist at the Australian National University, and I'm talking today as part of National Science Week 2021 and as part of a lecture series that was organized by the Australian Society of Plant Scientists on approaches that plant scientists from around Australia are using to safeguard our future food security. My talk will be on the research we're doing at the Australian National University to try and improve biological nitrogen fixation. One problem with agriculture is that it has a big footprint. The inputs into agriculture are huge and they're producing a lot of pollutions. For example, the annual emissions that are caused by agriculture around the world are estimated to reach 9 gigatons or more by 2050. And these emissions are caused, for example, by methane emission, by energy input into agriculture, manure management, but also in particular soil fertilization with synthetic fertilizer. And altogether, these emissions are estimated to contribute about 10 to 12% of all greenhouse gas emissions. So we need to do something about it. While nitrogen is really important for agriculture and agricultural production could not be competitive and productive without nitrogen fertilizer, farmers are using quite a lot of the stuff. So manure or synthetic nitrogen fertilizers are applied to crops at quite high amounts. They get washed into waterways where they could cause algal blooms and damage to ecosystems like coral reefs, for example. And also nitrous oxides emission are breakdown products from fertilizer that are volatilized into the air and are acting as very potent greenhouse gases. In addition, the synthesis of biological industrially produced fertilizers uses about 1 to 2% of global gas consumption. So this is a very high percentage that we need to try and reduce. Even just the economics of nitrogen fertilizers and nitrogen emissions are huge. So it's been estimated that just for the European Union, the cost of cleaning up nitrogen pollution is 100 to 400 billion US dollars annually. And this is both in terms of health costs because of nitrous oxides in the air causing respiratory problems, but also costs to ecosystems, in particularly to clean up the damage from nitrogen runoff in water, which is mostly a result of agricultural emissions, which are indicated by this green wedge, as is air pollution of ammonia and nitrous oxide. And to put that into perspective, the cost of cleaning up nitrogen fertilizers in Europe are bigger than the benefits of those fertilizers for farmers. So the problem with nitrogen is that there's very little in the Earth's crust, but a lot is contained in the atmosphere. 78% of our atmosphere is composed of nitrogen gas, but gaseous nitrogen has a very stable triple bond that most organisms cannot crack. And in order to use nitrogen, cells require it to be solubilized, for example, as ammonia, before they can build it into organic molecules. And this can happen either by industrial processes, so reacting nitrogen and hydrogen gas industrially with a lot of energy through the Haber-Bosch process, or it can be done by bacteria that can fix nitrogen biologically. And many of these bacteria form symbioses with plants. One of those, and probably the most important one, is the symbiosis of nitrogen-fixing bacteria called rhizobia with the roots of legumes. And the bacteria form these nodule structures on the roots of legumes like clover or peas, beans, lentils, chickpeas, soybeans. And they live inside these nodules that can be seen here in these little green dots that are the bacteria. And what they do in there is they basically are like a little nitrogen factory. They take nitrogen gas from the air and convert it into ammonia that the plant can use as nitrogen fertilizer without the problems of producing additional unused fertilizer that washes off into the groundwater. This symbiosis is very important economically. You can see here are some soybean plants that can form this symbiosis naturally. If they lose the ability to communicate with bacteria and form the symbiosis, you can see that in the absence of nitrogen fertilizer in this field, the plant does very poorly without these bacteria. In Australia, the amount of nitrogen fixed by bacteria in legume crops is about 2.7 million tons, and this is estimated to save about 3.4 million tons of industrially produced nitrogen fertilizer, with an economic value of about $4.3 billion a year in Australia alone. So the economy of this symbiosis makes sense. And there are two problems. So, and two questions that the international community is trying to solve in this field. One is that many species of plants, most species of plants cannot form symbioses like this. Only legumes and a related clade of plants that include, for example, casuarinas, can form nitrogen-fixing root nodules. And people are trying to find out why they can form it and why other plants like wheat or rice or corn or most of other staple plants cannot form the symbiosis. And this question has not been answered yet. But an equally important question is why legumes, for example, differ very much in their ability to form efficient or less efficient symbioses. And answering these questions is really important for breeding of new plants that can grow with much less synthetic fertilizer and benefit more from the ability of these nitrogen-fixing bacteria to colonize their roots. To understand that, people have been trying to understand the cellular and molecular processes that occur when the roots interact with these nitrogen-fixing bacteria. One of the important factors in the symbiosis is the chemical communication between roots and rhizobia. Plants produce lots and lots of chemical compounds. In their roots, for example, they produce flavonoids, which are these beautiful fluorescing molecules that are then transported into the soil where they activate the bacteria to initiate the symbiosis. The second essential step is that the bacteria have to get inside the plant without being recognized by the plant immune system that could evict them and stop the symbiosis. And here you can see how the bacteria get into the plant. This is a photo taken by one of my PhD students, Angus Ray, who developed this beautiful technique for visualizing the structures here in pink called infection threads that bacteria form when they invade the plant. So in green, you can see a single root hair cell that is infected by rhizobia, and you can see the bacteria travel to the inside of the root in these infection threads where they're safe from the immune system of the plant and from competition of other bacteria to be delivered to the inside of the root. And at the same time, the inside of the root, which you can see here in an optical section, is activated by these bacteria, by specific chemical signals from the bacteria that initiate new root organs. The nodules, this is the earliest stage of a nodule where the bacteria have reinitiated cell divisions in the root to form a nodule. And at the same time, they're basically telling the plant to send carbon supplies to these growing nodules to provide them with enough energy for the nitrogen fixation. So we're trying to understand how that process works and how the plant controls it. One thing that we've been able to do to improve nitrogen fixation and nodulation in legumes is to manipulate the signaling molecules that the plant produces and that it uses to communicate with bacteria, because we've exploited the fact that some of the same compounds that the plant produces also play a role in protecting plants from invasion by parasitic nematodes, which are a very, very big problem in agriculture, as well as from fungal diseases, which affect all crop plants and cause huge costs to agriculture. And by manipulating these signals in the root, we've been able to show that, for example, we can improve nitrogen fixation and nodulation in the roots of legumes. At the same time, the manipulation of these signals in the plant genetically will reduce the attack from parasitic nematodes, which you can see here in pink, invading the root. So these ones are dual approaches for balancing nitrogen fixation with pathogens, which also are a huge cost to agriculture. With that, I want to thank you for listening, and I want to thank various sponsors who have kindly supported our work on this. Bye-bye.